In this video, we will discuss about Oncolytic Herpes Simplex virus cause cancer-associated fibroblast subtype differentiation. So far, we got to know that this Oncolytic Herpes Simplex virus cause subtype differentiation in this 1:10 mixture model more than the KPC mice model. So from that, they have come to a hypothesis that this oHSV cause this cancer-associated fibroblast subtype differentiation that can be validated by checking this oHSV effect on the pancreatic stellate cells. So they did the in vitro treatment of this pancreatic stellate cells with this oHSV to see whether it will cause the CAF subtype differentiation or not, as PSCs has more CAF markers compared to the KPC. So in the mock, the results shown for these pancreatic stellate cells, and when they are treated with oHSV, we can see the cytopathic effect, and which is evident with this GFP fluorescence. And when they compared this cytopathic effect at different viral titer with different cell lines, we are able to see that for the pancreatic stellate cells, in order to achieve the same cytopathic effect, it needs around 1 to 2 magnitude less amount of this oHSV virus. So this indicating that the pancreatic stellate cells, since it has more CAF markers, so this oHSV can work efficiently on this CAF compared to this KPC and Dero, which has less CAF, compared to the PSCs. Thereafter, they also checked the myofibroblast and antigen presenting cancer-associated fibroblast, along with this induced cancer-associated fibroblast. So in these PSC cells, they are able to see a decrease in the myofibroblast, which is the tumor. This myofibroblast helps in this tumor proliferations or immune cell suppressions, whereas antigen presenting cells helps in the tumor suppressions, and immune cells helps in immune cells to detect the cancer cells. So it goes up. And since, however, there is no change in the induced cancer-associated fibroblast, the markers for this ICAF gone up, that is IL-6, and the tumor proliferation markers, that is TGF-beta, gone down in this oHSV treatment. So this all indicates that this oHSV will work better on this cancer-associated fibroblast, which is predominantly found in these pancreatic stellate cells. Thereafter, they checked the results with this KPC mice model and the MSC1 mice model using flow cytometry in the in vitro conditions, and what they are able to see for the KPC mice model, only there is an increase in this MSC1, whereas co-stimulatory domain or co-stimulatory factors, that is CD86, CD80, CD40, they do not see for the CD80 they do not see significant changes, but for the CD86 and CD40 the significant changes are there. However, when they check the signals for the similar markers, that is immune suppressions marker, tumor suppression markers, that is MSC1, and the proliferations markers, co-stimulatory markers, not proliferation markers, co-stimulatory markers, that is CD86, CD80, CD40, they are able to see that increase in this both MSC1 and MSC2. However, and also the CD86 and CD80, and they do not see significant changes in the CD40 between the mock. However, total amount is still higher than what they have seen in this KPC treated with oHSV. And when they compare the ratio of this antigen presenting markers and the co-stimulatory markers in the pancreatic stellate cells treated with oHSV and the KPC cells treated with oHSV, we are able to see that the relative expressions in the pancreatic stellate cells markers are higher compared to the KPC cells treated with oncolytic herpes simplex virus. So this in all together indicate that this oncolytic herpes simplex virus can cause more cancer-associated fibroblast subtype differentiation and compared to other cells. So this is what they conclude on the results that are presented in this figure. This is how they conclude on the results presented in this figure. If you need to understand more about these experiments and this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.